Hi, I'm Katharina from the Computational Health Department of the Helmholtz Center Munich. I will talk about design and power analysis for multi-sample single-cell transcriptomics experiments. For this, we developed an analytic power analysis method called SE-Power. We covered two groups of multi-sample experiments with our power analysis. EQTL studies, where we look in a population for significant associations between genetic variants and gene expression, and differential expression analysis in general, where we look at significant differences between two groups in a population, for example, disease patients and healthy controls. In both cases, single-cell RNA-seq can be used to identify cell-type-specific differences. These might be missed in bulk if we look at the whole tissue at once. So these datasets are really valuable to capture all existing differences and additionally then provide information of the cellular context. However, it requires to measure single-cell RNA-seq in a whole population, so it's even more important to perform a power analysis before and estimate the best possible design to then save resources, such as costs. That's the workflow of our tool. It's an analytic method which makes it efficient both for computational time and memory, and we try to make it as flexible for the user as possible, here shown a bit with the different gray parameters that all can be adapted. We want to estimate the overall detection power here below in blue, and it depends mainly on the cost-determining factors: the number of samples we measure, the number of cells per sample, and the number of reads per cell. Also, prior information plays a role. For example, the effect sizes that we expect. We can take these either from previous studies or set it based on our assumptions. To then calculate the overall detection power, we split it into two parts. The expression probability, which says how likely it is to find a gene expressed. That's quite important here because we have single-cell RNA-seq, which is much sparser than bulk, and we already miss part of the genes. And then the DE or EQTL power, assuming that we detect a gene, how likely it is to find significant differences between the groups. To now start with the expression probability, when we combine these overall genes, we can estimate the number of expressed genes. When we looked at our own PBMC dataset, we saw that the number of expressed genes on the y-axis depends mostly on how many cells we measure per cell type here on the x-axis, and then the read depths here shown by the different colors. We say that a gene is expressed if it has a certain number of counts in a certain fraction of individuals, and both parameters can be chosen by the user. So we look at the expression on an individual level instead of a cell level. The approach that we use here is also known as Pseudobulk. We aggregate the counts for each gene per individual and cell type to such a three-dimensional matrix. The approach has also been shown to be one of the best performing for individual level DE studies using single-cell RNA-seq data, and is also very commonly used for EQTL studies. So the goal is to estimate now the expression probability here, and we do this based on an expression prior that captures the general cell type specific count distribution. So we start from a count matrix from a training data set, and we estimate for each gene and cell type the specific count distribution. We then generalize it depending on the number of cells and the read depths. And with this, we can estimate the expression probability for an unknown distribution in the planned experiment. For the mathematical details, please contact me or have a look into our preprint. We use our expression model now to predict the number of expressed genes in two different datasets. In the top row, you can see the results for our PBMC dataset that we used to generate the expression priors. The dashed lines, which show our estimations, match very nicely the solid lines, which are the observed counts. But more important, also the prediction in an independent dataset works well that was generated in a completely different setting. So we can be confident that our model works. With this, we come to the second part of our model, the DE and EQTL power analysis. We can use here methods developed for bulk if we input the mean and dispersion parameter of our expression probability model. We correct also for multiple testing using again the expression probability model to estimate the overall number of expressed genes, which is the number of tests. We can then combine both to the overall detection power by building the product, as both are conditionally dependent on the mean and dispersion parameter. In this example, you can see nicely that the number of cells is the major determinant for the expression probability, while the sample size is in both the DE and EQTL case mostly important to calculate the power. And then we have the overall detection power as a combination of both. We benchmarked our model against different established simulation-based methods, and we saw that both match very nicely. Each point is here a different parameter combination. The big difference is the memory and runtime requirements. We have an analytic method which is much faster and also more memory efficient. To make the best use of our efficient calculations, we now optimize the parameters for a fixed budget. So we add additional cost function, and then we have a grid of possible parameter values. Here it's number of cells and readouts, and the third parameter, here the sample size, is defined always uniquely given the combination of the other two and the overall budget. We see here that the optimal parameter combination is shallow sequencing of a large number of cells. And similar results we get in a lot of different scenarios when we evaluated our power analysis for different budgets and also different priors, for example, different effect sizes. I hope I could convince you to test our power analysis to yourself before your next experiment. It can be used for both droplet and plate-based technologies. Please contact me with any questions or suggestions. I'm looking forward to discuss with you.